Carbs are mine too when I really want to have some pizza, because I like pizza. I think if I could only have one type of food for the rest of my life, people have asked me, everyone's like, pizza, pizza. I said, no, peanut butter and jelly sandwiches. I could definitely do that. Oh, you're from New York? Huh. All right, fair enough. Fair enough. I was looking for something here, but let's talk about proteins. Pretty cool. A pretty cool classification. All right. For macromolecules. And when you look at this, I'm going to give you a second. I'm not going to say anything for a few moments, but I want you to look at this list and see all the functions that proteins have. All right. And we're going to pretty much cover almost all of those. Okay. Almost all of those this semester. Okay. So the first one there, when we talk about functions for a protein, catalyst, okay? Chemical reactions occur in your body, happens all the time, all right? So any type of metabolic reaction that occurs inside the cells, you're going to need a little help in getting it started. Just like with a fire, you want to get a fire going, you need a little help to get it started. You can use a lighter. A lighter is your catalyst. A lightning blast from a storm, all right, can be a catalyst. That's what proteins, they're called enzymes, but they're this catalyst for a lot of the metabolic reactions in our body. You hear a lot about this next function in the news because we always are talking about COVID-19, it seems. Well, people talk about getting tested, and then you'll hear terms like antibodies. Antibodies are proteins. So your body, specifically plasma cells, will build these antibodies, and these antibodies will attack and destroy germs. Okay. Transport, we'll talk a little bit about that in chapter 4, about how cells, especially cells, can move things in and out of the cell. All right. Also, we have in the cells what we call our cytoskeleton, and the cytoskeleton gives the cell its shape, and so that cytoskeleton is made from proteins. Movement. All right, you have your actin and myosin proteins, which are called myofilaments, and what they do is allow the muscle to contract and relax. Regulation, there's a lot of different types of proteins that are going to allow for certain types of regulatory functions within our cells and our tissues overall, and we'll get some detail about that in chapter 4. And then finally, storage. And we'll talk about, like I said, we're not going to really talk about too much about storage this semester. That'll be more into 11. So we saw with sugars, or excuse me, with carbohydrates, remember monomers, not monomers, excuse me, monosaccharides, which is a simple sugar, and then we had a disaccharide. We take two monosaccharides, stick them together, and then we have polysaccharides. Well, a monosaccharide is what we call a monomer. Okay, so when we were making glycogen, we were just making a chain of all these monomers of glucose and sticking them together. Well, proteins are similar in that they have monomers. All right, but we call those monomers amino acid. So we're going to learn here when we're building up, when we're adding amino acids to each other, all right, building up, make a big long strand of them. We're eventually going to get, if we get it large enough, a protein. Well, think about it. Digestion does the exact opposite. So when you eat something like a steak, something protein rich, okay, and you're digesting it, you're doing the exact opposite. You will start to break those proteins down back into their amino acid monomers. All right, so we're going to talk about anabolic metabolism, which is building things up, okay, and then in some of our chapters here, you'll learn about catabolic metabolism, which is tearing things down. Okay, so in us as humans, there's 20 different amino acids. All right, and we have two categories for them, two main categories: essential and non-essential. All right, so some amino acids you can get, all right, from your diet. Your body can't produce them, and then some of the other amino acids your bodies can actually make. Well, each amino acid has a functional group on each end. All right? One functional group is called the amine group, and the other functional group is called the carboxyl group. Okay? And so what we'll see is that both of these groups are linked specifically through covalent bonds. Does anyone remember what type of a bond a covalent bond is? Yes, beautiful. That's right. You are correct, Heather. They share electrons. Sharing is, and that's what covalent bonds do. They share with one another. Okay? So we'll see in amino acids that we have a carbon atom there, and we have an amine group attached to it and a carboxyl group attached to it. And you're probably thinking, well, what's all that? I'll show you pictures. Don't worry, I got pictures. I got pictures, people. All right? So I will show you those. Okay? All right. So when we're talking about also with that carbon that I just mentioned, right? It also will be bonded to a hydrogen ion, excuse me, a hydrogen atom, right? And then it has a side chain, okay? And it's different from amino acid to amino acid. So we call that an R group, okay? So I won't go through the names of all the different amino acids, but we got leucine and valine, which are two types of amino acids, where they're going to have different R groups from one another, okay? And that's how we're able to distinguish one amino acid from the other, okay? Let's see, let me pull that up. I'm going to come right back to the slide. All right, so here we can see our amino acid. Whoops, wrong way. So here's your amino acid. Here's your carbon. The hydrogen group is attached to it. Here's your amine group. Amine group is the one with the nitrogen in it, and it has two hydrogen atoms. All right, and then we have the carboxyl group, or the carboxylic acid. All right, and it's going to have its own carbon, two oxygen, and a hydrogen. Okay, and then we have our side chain, our R group. And again, it varies from amino acid to amino acid. All right, so keep that in mind. All right, so onward with the proteins and the amino acids. So when we are on our way to try to build a protein, okay, we will start to attach amino acids to one another. And we do so by using a special type of covalent bond, and that's referred to as a peptide bond. So the reaction when we're sticking these two amino acids together, the process of this covalent bonding, all right, we refer to that process as dehydration synthesis, and it's a chemical reaction, okay? So what happens is the amine group of one amino acid is going to interact with the carboxylic acid group of the other amino acid. And because of that process, all right, or that reaction, all right, the amine group is going to lose one of its H atoms, okay? And the carboxylic group, the carboxylic acid group, is going to lose its OH molecules, okay? So as you can see, we have two H's and one O. Well, that's H2O, okay? So the actual product that comes out of this reaction, all right, is going to be water, okay? So now we have two amino acids attached to one another. I'll show you a picture here. So on one end of those attached amino acids, we'll have what we call the free amine group. That means that that amine group is not attached to anything. So we call that the N terminal. And then on the other end, we'll have a free carboxyl group, and that's the C terminal. All right, I'll come right back to the slide. Back to my picture. So that's what we're seeing here. Okay, so you have one amino acid and another amino acid. Okay, so we're going to attach, all right, this carboxyl group here and this amine group here, okay, to one another. And as that process occurs, we lose this hydrogen and this hydroxide, and we get water as a result, all right, and we get, this is, and they both will connect to one another. So that's what we'll see. That's what we'll see here. This is actually a polymer protein. We're just adding a whole bunch of amino acids to each other, but you can see where this is a peptide bond. All right, so for this to occur, this reaction up here had to take place. Okay, and we call that, all right, our dehydration synthesis. Well, think about it. Dehydration has to do with water. Usually it's a low water content in us. Well, hydration has to do with water. So just think, when you see dehydration synthesis, we're going to get water as a product from that reaction. Okay. So here's our picture, and you can see, all right, what we do is we're just taking all these amino acids, okay? Those are our building blocks, our monomers, and we just start to stick them onto one another. Here's a peptide bond. That's what this is representing. That's a peptide bond. And that's that covalent bond between amino acids. And we just start to stick them on. All right, so you have a free end here, all right, on our polymer. And that is, and if it's an amine group, we call that the N terminal. If it's our carboxyl group or their carboxylic acid, it's the C terminal. Okay? All right. So, in order to be classified a protein, you have to have more than 200 amino acids strung together. Okay? If you have 199, you're not a protein. If you have 200, you're not a protein. If you have 201 amino acids strung together, you're a protein. Okay? So, if we string 3 to 20 amino acids together, we call it an oligopeptide. And then if we get 21 to 199, we have a polypeptide. Do not ask me where they got these numbers from or where this comes about, okay? But that's just how it is, okay? So when we get our protein, our body will attach things to it, all right? Add things, maybe take some things away. Well, if our body adds a carbohydrate group to it, we now refer to it as a glyco, sugar, protein, a glycoprotein. And these glycoproteins you'll see on the surface of some of our cells. We'll use them as cell markers. Okay, and an example that we give here are the glycoproteins on our red blood cells, blood typing people. So if you're an A positive, a B negative, all right, O, all right. Well, in that case, if you're O, you don't have any surface antigens, all right. But regardless, all right, when we're blood typing people, it's because of these specific carbohydrate proteins, these sugar proteins, these glycoproteins that sit on the surface of the cell. All right, what are the monomers for proteins? You need to know this. I've seen this asked on test questions before. All right, you need to know that a monomer for a protein is an amino acid, and they're linked together by this peptide bond, all right, which is a type of covalent bond. It's a sharing of electrons. Good so far? I hope. All right, sorry to be OCD, but what if there are 200? Yeah, I know, exactly. If you're 200 amino acids strung together, you're a polypeptide. You have to be greater than 200. So 201 and larger, then you're a protein. If you're less than 200, all right, if you're 199, you are a polypeptide. All right, I'll go back to that slide there. Okay. Oligopeptides, 3 to 20 amino acids. A polypeptide is 21 to 199 amino acids. Oh, I see where you're probably, this should be 200, sorry. So that more than 200 amino acids, then we have a protein. All right. I'm drinking, normally when you guys see me in class, I drink regular water from school, but at home, I'm drinking sparkling water, black cherry coffee. Yummy. All right, so when we categorize amino acids, all right, we do it based on their R group, okay? So there's two types that we're going to talk about. we're going to talk about nonpolar and polar. So keep this in mind, all right? When we're talking about nonpolar amino acids, all right, the R group, okay, is going to contain hydrogen ion, excuse me, I always want to say hydrogen ions. I'm so used to saying that in this chapter. Hydrogen atoms only, or hydrocarbons. What is a hydrocarbon? Does anybody recall what a hydrocarbon is? No? Well, a molecule with only hydrogen and carbon. That's correct. So the R group for nonpolar amino acids are going to have either just hydrogen or a hydrogen and carbon. Okay? All right. So these types of amino acids are going to group together with other nonpolar amino acids. The R groups it's like a side chain. Okay? It's what differentiates amino acids from one another. So amino acids have pretty much the same molecular skeleton except for the R group, which is this one chain of molecules that come off the side of it. And they're different from one another. And that's how you're able to differentiate. They have different properties because of the other groups there. Yep. All right. So they're going to interact with other nonpolar amino acids, like sticks with like in this case. All right. But it'll do so through hydrophobic or water-fearing interactions. All right. Because remember what I was talking to you about, things that will dissolve in water and things that won't dissolve in water. All right. Things that won't dissolve in water are going to be what? Nonpolar. Correct. That's right. Yes. Things that will dissolve in water will be polar and ions. Okay. So our polar amino acids, okay, same, all right, they'll have their R groups will have other elements besides hydrogen or hydrocarbons. So they can be a little bit more diverse. Okay? They could have nitrogen, for example. All right, they can have other items other than, all right, hydrogen or the hydrocarbons. So again, they'll have interactions with other polar amino acids, all right, and they'll also interact with water because they are polar. So they can dissolve in water, all right. So they can interact with other polar amino acids and they can also interact with water because that's how they roll. All right. Two other ways that we can organize amino acids by their R group, okay? The charged amino acids, that means that the R groups can either have a negative or a positive charge. So what we'll see here, all right, these types of amino acids can form ionic bonds. You guys remember what ionic bonds are, right? Correct, it is. It is different than polar, okay? I don't want you, I know it's easy to do this, but I don't want you to think when I use terms like polar, okay, that it has strictly to do with a positive and negative charge. Because remember, I mean, the Earth has a north and a south pole, so it has polarity, we call it, okay? And an amino acid has polarity, meaning you got one end that has the the amine group, and the other end has the carboxyl group, okay? Polarity is a pretty broad term. Okay, so ionic bonds, all right, are the type of bond in which one element will donate an electron to another, and one element will accept an electron from another. Okay, whereas covalent, sharing, ionic, you gain or lose an electron. Okay, all right. So again, these guys here are going to be hydrophilic. And then finally, we have some amino acids that have special functions. For example, proline, all right, it will cause a bend in the protein chain. And again, that's more of a chemistry, organic chemistry situation. But when that occurs, when you alter the molecular, the structural formula, all right, of certain compounds, you'll influence some of the functions that they'll have. Okay, and you'll see here, like with cysteine, you can get a disulfide bond, all right, which means, all right, you'll have a molecule that has two sulfur that will bond together. This one's kind of important, all right, when we talk about translation and transcription and the making of proteins. So when we're taking our DNA and reading our DNA, and that DNA says, hey, I want you to make a specific type of protein, make an insulin protein, all right? So anytime you're going to make a protein, you have to start off with a specific amino acid, all right? And it's basically like the in a race, all right, in a car race, they always use what? The green flag. So methionine is the green flag in making proteins. It's the first amino acid that is used, or that is being made, all right, and its genetic code is AUG. For those of you that have any experience with genetics or some biochemistry there, okay? That's all you're going to really need to know with that. But anyways, this whole point of this slide, or this part of the slide, is just to tell you that these are amino acids with special functions. They're spatial. I won't go over this in detail, but this is just a picture of all these different amino acids that I just talked about, okay? All right. Let's talk about our nucleic acids. We're not going to go into a lot of detail here because we're almost done with this chapter, and I know a lot of you are probably like, thank God. All right. So when we're talking about nucleic acids, I want you to think of molecules or macromolecules that are going to store and transfer genetic information in our cells. Okay. Storage and transferring of genetic information. So we have two classes. We got DNA, deoxyribonucleic acid, and then RNA, ribonucleic acid. Okay? And I'm sure you've heard of at least DNA. Okay? So DNA is a double-stranded nucleic acid, and we find it, all right, in these structures called chromosomes that live in our cells. Okay? And these chromosomes will be found in the nucleus, which we'll talk about in chapter 4. That's the control center of our cell. And in the mitochondria, which is the powerhouse of our cell. We have actually, I believe it's 16 genetic, not chromosomes. Well, maybe I can't remember now. But anyways, the genetic material that's stored in your mitochondria, you get from your mommy, okay? So if you have any type of genetic condition or disorder, all right, that comes from a mitochondrial DNA issue, you can thank your mommy for that, okay? Because that all comes from mom. All right, ribonucleic acid is single-stranded. You want to know the difference? DNA is double, easy to remember. They both begin with D. All right, and ribonucleic acid, RNA, is a single-stranded. So we'll also find RNA in the nucleus, but we'll also find it in the cytoplasm of the cell. The cytoplasm of the cell is the goo that sits outside the nucleus and underneath, all right, the plasma membrane. Okay? All right, so when we talk about these nucleic acids, we're going to have some building blocks for those. All right, and those building blocks are called nucleotides. Okay? So a nucleotide, here's the ingredients for the nucleotide. All right, a sugar molecule, and it varies from nucleotide to nucleotide, a phosphate group, and what we call a nitrogenous base. Okay? So I need to point out to you the nitrogenous bases. We have 5 of them, okay? And I have them listed here. The first 2, all right, adenine and guanine, we call those purines, okay? And they have a double ring structure to them. Those are found in DNA. All right, the third one here, thiamine, that's in DNA also. All right, but these other nitrogenous bases, cytosine and uracil, those are in RNA only. But these 3 here, thiamine, cytosine, and uracil, all right, have 1 ring. And we call those 3 pyrimidines. All right. There's much more to know, not for this class, but to know about that kind of stuff if you're going into chemistry. But that's as far as we're going. I want to get to this one here. This is the last thing for chapter 2. Okay? This is a very important nucleotide. I want you to get very familiar with it because you're going to get intimate with it throughout the course. Adenosine triphosphate. Okay, triphosphate is describing to you how many phosphates, all right, are in, all right, this nucleotide. Okay, this is a nucleotide. So it has its nitrogenous base of adenine. It has a ribose sugar, and I'll show you a picture of this molecule. But what the really important part is this part right here, 3 phosphate groups. Okay, and these phosphate groups, all right, are bound to one another through covalent bonds. Okay, and the last 2 here, well, actually all 3 of them, but the last 2 are energy rich. So when you break a bond in between the 2 phosphate, you get released energy. And so this molecule here is crucial for energy transfer within the cells. Okay, and you'll hear me say terms like this is the energy currency of your cell. All right, so we have this chemical energy that's stored in this molecule. All right, and so this molecule can travel, within the cell. All right, and be used to help with chemical processes. Okay, so these other 2 molecules here, all right, NAD and FAD, okay, they help in making ATP inside of our cell. So if ATP is the American cash, all right, our currency, think of NAD and FAD as poker chips. And so when you win at the casino, hopefully, and you go to cash out, all right, you're going to go to this in the cell, you're going to go to the electron transport chain, okay? These molecules will, and they'll cash out to get ATP, all right? In real life, you go to, I don't know, the money exchange booth at a casino, and then you hand them your chips and you get cash, okay? But that's what they do. Their job is to help in the formation of ATP. So think of ATP as the chemical molecule that we use, okay, to make energy within the cells. So here is, all right, the molecule. Over here, you got your adenine, the nitrogenous base. It's a purine, so it's a double ring structure, okay? Then we have our sugar group, the ribose. It's a 5-carbon sugar. And then this is the part that I want to draw your attention to, the triphosphate. See, 1 phosphate, 2 phosphate, 3 phosphate. So these are energy-rich bonds right here, okay? So if I cleave this bond off, I'll get a release of energy, and I can use that energy for certain metabolic processes. But once I cleave that bond off, and this phosphate bond leaves, You had to ask me that. I'll go back. Flavin adenine dinucleotide. That's the one that is found in well, it's found all over the place. But flavin gives if you take a multivitamin, and especially if it has a lot of vitamin B2 in it, riboflavin, that's what gives your pea that really bright yellow coloring. You got it. All right. So we cleave this bond, we break it for some energy. That's right. Thank you, Stacy. Now we go from ATP to ADP, adenosine diphosphate. And now what happens if we cleave this one off? What do we call it? Well, what's one? What do we say for one phosphate? Monophosphate, AMP. All right. And you're going to be familiar because you're going to hear this. So when you see it up there in notes or in wherever, and you see ATP, ADP, AMP, all right, just know that we're talking about this molecule, all right, and it's lost one or two phosphate groups. All right, y'all, that's it for that chapter. Okay, we can celebrate now. Woo! I know. It's bittersweet. Bittersweet. It's a tough chapter, y'all. All right, let's jump right into just sweet. Yeah, it's not bitter. All right, let's jump right into chapter 4. We got to talk about this biology here of the cell. All right, so we're done with, all right, the atom and molecular and macromolecule portion. Now we're starting to increase in our organizational chart here. Okay, so as we start from the simplest and get to the more complex, we're moving up a level now. Now we're going to cells. All right, so you definitely want to know, all right, that cells are the functional units of our body. Okay, functional units of our body. So you'll find out if you have a pretty decent understanding of cell biology that it'll help you to understand more of the larger overall system physiology functions and anatomy functions. Because when we get into tissue, we'll start to kind of explain that on a little bit of a larger scale. Okay, but for now, we're going to focus on what a cell is, what the components of the cell are, and some of the more important biological functions that cells undergo. Okay? All right, but when it boils down to it, the functions of the cell, of each individual cell, are really important for the overall physiology and anatomy of us as an organism altogether. So there's 3 parts, all right, to a cell. We have the outer part here. Yeah. I'll go back real quick. I'm actually looking here really quickly at what I'm drinking. And I wanted to make sure that there wasn't any aspartame in there. There's none, just in case you were wondering. There's a little bit of caffeine, though. It's actually quite tasty. All right, so our 3 features, all right, for a cell is 1, the outer envelope, the plasma membrane. This is very important, and there's a reason why it's underlined, all right? It is a limiting barrier. I've been there, Lexi, I've been there. So plasma membrane is a limiting barrier, all right, that is going to separate the inside of the cell from the external or outside environment. And so that limiting barrier is going to let certain things in and let certain things out. You don't want it to let everything in and let everything out. I mean, do you do that at your house? Do you just let anybody into your house that wants to come into your house? No, heck no. Well, your cell does the same thing. So we're going to let certain things in and out of it, okay? Kind of like a filter. You're going to find out No. Well, simple squamous is a type of cell, but this is going to what we're going to learn here in this chapter is going to apply to simple squamous, cuboidal cells, columnar cells, transitional cells. This is general for all those tissue cells that we talked about in lab on Tuesday. Okay. So remember what we talked about when we talked about the modified extensions in lab somewhat? We talked about cilia and the microvilli, okay? And there's three types of modified extensions of the plasma membrane, okay? The cilia are like those hair-like projections that help to move things by the cell. The flagellum is really big. It's like a tail, a big tail. And that actually moves the cell. There's only one cell in the human body that has the flagellum, and that's the male sperm. No other cell in the body has a flagellum. So that's an easy one to remember. I've seen that asked as a test question before too. So keep that in mind. Sperm, only cell in the body in humans that have flagellum. And then we saw the microvilli. Remember the brush border? Okay. The purpose of the microvilli is to increase the surface area, okay, of a cell so you can improve absorption. All right, the second structure is the nucleus, the control center. Okay, it's the largest structure in the cell, and it also has its own nuclear envelope. So like how a cell has a plasma membrane surrounding it, well, the nucleus has something similar to its own plasma membrane, and we call that the nuclear envelope. Okay, and so inside the nucleus, we have nucleoplasm, which is similar to the cell's cytoplasm. But this is where we keep our DNA. It's like our safe, our vault. We lock away our DNA, all our genes, our human genome is stuck in our nucleus there. Okay, and we only bring it out when we need to. And the third feature of the cell is going to be the cytoplasm. That's the goo that's inside the plasma membrane, but outside the nucleus. Okay? And there's three components to our cytoplasm, and we're going to go through each one here. All right? We'll talk all about it. We got our cytosol, the organelles, and the inclusions. Cytosol is kind of like, it's a viscous liquid, kind of like, I'm going to say motor oil, all right? But that's like the liquid portion of the cytoplasm. You know how we have organs inside of our body? Well, a cell has its version of organs inside of it, and we call those organelles. And then the inclusions are everything else. But they're very, very temperamental. They come and go. And I'll explain to you what all that is. We're going to on this next slide here. All right, so let's start off with cytosol. Okay, first of all, you need to know this term here for it, intracellular fluid. That's another name for the cytosol, because you'll hear in terms when we're describing, especially with neurons, you'll hear me talk about extracellular fluid and intracellular fluid. So it's real simple. Intracellular fluid is all the fluid inside the cell. Extracellular fluid is all the fluid outside the cell. Okay? So we have tons of water here, very high water content in the cytosol. So certain things like to dissolve into water. Remember, polar and ions. So we'll have dissolved macromolecules with polar properties and ions. Makes sense, right? So that's what we'll find in the cytosol. All right. The second component to our cytoplasm are the organelles, which stands for little organs. Okay. Now we'll talk about these in a little bit here in our slideshow, but they have various functions and various configurations or shapes. So we have two categories for them. The first one is a membrane-bound organelle, and that essentially means that this organelle will have its very own capsule around it or membrane. Okay, because if it has to do certain jobs, it needs to kind of seal itself away from its immediate environment. Okay, and then you have non-membrane bound organelles, right? And they don't have a covering or a capsule around or a membrane around themselves. So it doesn't matter if they're surround whatever's going on in their environment that's immediately surrounding them. All right, so 3 components: cytosol, organelles, and our next one here. Oh, well, sorry, I'm jumping the gun here. Out of the organelles are 2 membrane bound, or membrane and non-membrane bound. We have the membrane bound have the organelle, and we're going to go through each one of these. The endoplasmic reticulum, one of my favorites, the Golgi apparatus. Well, actually, all these, I love all these. Lysosomes, peroxisomes, mitochondria, those are all fun. And then our non-membrane bound, all right, they do not have a membrane, but they are made of protein, all right, our ribosomes, the cytoskeleton, all right, a centrosome, we'll talk about what that is, and then one of the really cool ones, a proteasome. All right, don't worry about writing we're gonna go through all these anyways. Okay. So now the third part of our cytoplasm are the inclusions. Okay, these are the structures I was telling you about that come and go. All right, they're only inside the cytoplasm for a short period of time. Okay, so we'll add to the cytoplasm and we'll take away from the cytoplasm. All right, perfect example we've talked about is glycogen. Okay, when it's time for your liver to start to break down the glycogen for while you're sleeping in the middle of the night, all right, because it's stored right there in the cytoplasm. Triglycerides, the same thing in our adipocytes. Okay, like well, salts again. That's going to be they'll dissolve because remember what happens to salts with in water. You get salt water. So in the cytoplasm, they'll dissolve and mix into, and most times, your cell, and you'll get to learn about this, neurons especially, but a lot of cells will want to split the salts apart because they like to use sodium exclusively. They don't need a sodium chloride molecule. They'll want sodium, and they also want chloride. So what will happen is salts get broken down quickly, and then they utilize those ions for whatever biological purposes that they want. All right, you want to see a cool picture of the cell? There it is. Let me blow it up a little bit real quick. All right, so you can see here, all right, we have our plasma membrane, okay? And the structures on it, here's our microvilli. You can see how it's an extension here of the actual plasma membrane. Okay, and that's just helping us to increase our absorption surface area here. There's the flagellum right here, this big long guy. Okay, and then the shorter hair-like structures, those are the cilia. See, the next structure here, well, this is a generalized cell, but yes, the sperm cell is the only cell that you'll see a flagellum on in the human body. Okay. This here is our nucleus, big structure. You can see it's got its own nuclear envelope, its own version of the plasma membrane. And on the inside, you can see all the genetic DNA material. These are the chromosomes here. And then inside the nucleus, you got this structure here called the nucleolus. More on that. We'll talk more about that. Then you can see all the various organelles inside. Here's the mitochondria, another mitochondria. No, you will not have to label this on a test. No. All right, so we're going to go through all these various different organelles here. All right, let's talk about a couple of the features and some of the functions of a cell. All right, so some of the functions that you'll notice about a cell, and we take it for granted when I was going over the different types of cells in lab class on Tuesday, but you notice how squamous cells and cuboidal cells and columnar cells had specific shapes. All right, well, believe it or not, that is one of the functions, all right, for these cells. all right, is to maintain both the integrity and the shape of the cell. Now, the integrity, what we mean by the integrity means like the strength of the cell, all right? If you have a cell that's doesn't have a lot of integrity, it doesn't mean it has poor character traits or anything like that. It just means, all right, that it might not be strong enough to support itself, okay? So a lot of this is dependent upon the plasma membrane. All right, when we talk about the internal contents, we're talking about things like the cytoskeleton, all right, which we'll get into, okay? Well, a cell is a living thing, so of course, living things have certain biological functions going on, all right? And during those biological functions, it needs energy. So it needs to obtain nutrients, okay? And depending on what those biological functions could be, all right, if it's to make something, if the cell gets damaged and it needs to repair itself, whatever, okay? It will need to use those nutrients to help form these building blocks for whatever its function is going to be. So essentially, it needs to get this energy to live. If it doesn't, it's done for, okay? All right. Well, whenever you have biological functionings going on, chemical reactions, biological reactions, you're going to have waste products. Like when you're driving your car. To get your car to go, you put gas in it. That's your energy source, your nutrients for your car. All right, but your car is giving off exhaust. Cell does the same thing. All right, so it needs to have, now if you didn't have an exhaust pipe on your car, where's all that exhaust? I can't talk. Where is all that exhaust going to go? All right, it's got to go somewhere. Well, the same thing with your cell. It's got to get rid of the waste that it's generated from all these processes that are going on. If it doesn't, they'll build up inside the cell, damage the cell, and quite possibly kill the cell. All right, so it needs to get rid of all that waste accumulation. All right, now some cells, not all, but some, all right, want to reproduce, or what we call cell division, when one cell divides into two. Okay, they're going to make more of themselves. All right, certain cells won't do that. A neuron is an example of a cell that will not divide. We call those types of cells amitotic. Okay, if you can read that. Amitotic. All right. So the reason why some of these cells divide is one, because maybe they've been damaged, or you're growing, okay? And when you're going through puberty or growth spurt when you're younger, all right? Correct. Well, there's two types of cell division. They cannot divide. There's two types of cell division. In fact, I'll type it in here. All right, there's mitosis. I'm going to make sure I spell this right. All right, and then there's meiosis. Oh, you beat me to the punch. Yes, that's correct. All right, mitosis and meiosis. Okay, one has to do with sex cells dividing, and the other one has to do with all other types of cell division. And we'll get there. We'll talk about it. I like to see you guys thinking, though. I like it. All right, so the main three structural features of a cell, you need to know those three. Great test question. Plasma membrane, nucleus, and the cytoplasm. All right, very important to know. All right, so what we're going to do now is we're going to talk about, all right, our organelles. And so I'm going to start off, all right, with the endoplasmic reticulum, the membrane-bound organelles. Okay, so if you look at our picture here, our endoplasmic reticulum is in close proximity to the nucleus. Okay, so when it's time to make things, all right, proteins, the information, the blueprints are inside the nucleus. All right, so we got to get the blueprints out of the nucleus into an area so we can start making whatever's on those blueprints. And our DNA hold the blueprints. So we got to get the DNA from inside the nucleus to outside. So think of the endoplasmic reticulum, and there's two types of endoplasmic reticulum. You can see this type over here, All right, it's kind of bumpy. Okay, so we refer to that as the rough endoplasmic reticulum. These little bumps are ribosomes. Those are just proteins. We'll talk about those in a moment. And then you got a smooth endoplasmic reticulum. So essentially, an endoplasmic reticulum is just a tubular network, okay? And it's close to the nucleus there, okay? So when we talk about the endoplasmic reticulum, there's two types. We'll start with the easier one first, okay? The first one is the rough endoplasmic reticulum, okay? So we'll have these these ribosomes in the rough endoplasmic reticulum that will make proteins, all right? And they're going to send these proteins to either this plasma membrane or send them to be exported out of the cell. So you'll see a lot of rough endoplasmic reticulum, all right, in cells that need to make a lot of protein. Perfect example is the beta cell, which is in the pancreas, and that's the cell that makes insulin. All right. So we're also talking about peroxisomes, but they're also made in the rough endoplasmic reticulum. And more on peroxisomes in a little bit. The other type of endoplasmic reticulum is the smooth endoplasmic reticulum. All right. And it's got a couple functions itself. It's going to make stuff, transport stuff, and store stuff. Lipids, for example. All right. Phospholipids. Does anyone remember the significance of phospholipids? Where do we see a significant amount of phospholipids? In the plasma membrane. The plasma membrane. So it's good. Smooth endoplasmic reticulum, that's where you have a factory, a right on-site factory for a structure that you need to have a lot of, because there's always going to be damage to the plasma membrane. You got to fix it, you got to repair it, you got to add stuff to it, you got to take stuff away. So the smooth endoplasmic reticulum is great because it will make the phospholipids. It'll even transport the phospholipids over to the plasma membrane for you. All right? So if we got to make glycogen, what better place to make it than in the smooth endoplasmic reticulum? So you're going to see in the liver cells and in some of the skeletal muscle cells a significant amount of smooth endoplasmic reticulum so we can make that glycogen. This is very important, the detoxifications of drugs, alcohols, and poisons. Your liver is one of, a few organs that helps to break down drugs into an active form. A lot of times when you take a drug, for whatever reason, it's not in its active form, and it needs to get broken down and go into your bloodstream and then travel to either the liver or the kidneys or some other organ, and it'll undergo certain chemical reactions before it's activated, okay? But your liver also will help to break down dangerous drugs to you, okay? So the liver is a very valuable organ. For those of us that have experienced too much drinking, all right, the liver helps to break down alcohol as the blood is traveling to the liver. The liver helps to break down the alcohol. So it's the actual organ that helps to sober you up. Certain poisons too, all right? So the liver does a lot. We'll also see a lot of smooth endoplasmic reticulum in the testes because of the testosterone that's produced there. Yeah, it's funny. Every time I mention that, Gracie, in classes, people always bring that up. Cirrhosis of the liver. Now, cirrhosis isn't just caused by too much drinking, but yes, if people drink too much, all right, just like with anything, too much of anything can be bad. Smoking, drinking, all right, it can cause eventual permanent damage to the liver. And when the permanent damage occurs, it starts to create scar tissue. And the nice thing about the liver is that it can regenerate. I mean, in a healthy young liver, you can cut away almost 80% of the liver and it will regenerate itself. I mean, it's a fascinating organ. I just won't eat liver. You know, I mean, I don't want to eat something that has to do this function of detoxifying drugs and alcohol and poisons. And I'm not talking about a human liver, a liver in any animal. All right. And when I mean I'm not talking about a human liver, I'm talking about I won't eat human liver. I'm like Fonzie. If anyone knows Fonzie from Happy Days, he did not like liver. All right. This slide here basically shows, it's just a repeat of what I just showed you. Okay. So again, I'm not going to spend much time on here. Just pointing out that with the rough endoplasmic reticulum, all right, we have those ribosomes, all right, that are on the endoplasmic reticulum, okay? And what they'll do is they'll help to make proteins, and then what will happen in the rough endoplasmic reticulum, some of these proteins will be put into these membrane sacs because they're going to send, all right, those proteins to this structure here, the Golgi apparatus. And I'll talk to you about that in a moment, okay? All right. Golgi apparatus. This is the warehouse of the cell, meaning this is where proteins will come, all right, and get modified, and they'll get packaged up, and they'll, like I said, they'll either get sent to the plasma membrane to be incorporated into the plasma membrane, or they'll be sent outside the cell. So the Golgi apparatus, there's actually a song about this organ, all right, organelle, excuse me, and it looks like it's a bunch of these long sacs or elongated sacs that sit on top of one another. And there's that term again, polarity. Okay, it doesn't mean there's a positive and negative charge. It just means that there's one end to this structure and then there's another end to the structure that's got a certain name. So the cis face is going to be closest, that's the fatter end usually, is going to be closest to the endoplasmic reticulum. And the trans face is going to be at the top or closest to the plasma membrane, or distal to the endoplasmic reticulum. Don't worry, I'll show you a cool picture on the next slide. Much better than my horrible drawings. I'm sorry I have to subject you to my bad drawings, but we all got a deal. All right, so again, big function for this structure, all right? It modifies proteins, it's gonna pack these proteins, it's gonna sort them out, send them to where they need to go, all right? And to do that, it creates what we call secretory vesicles, okay? It's like a bubble, all right? Bubble that you see floating through the sky when you get soap and water together, okay? So these secretory vesicles, these bubbles, will go up to the plasma membrane and merge with the plasma membrane, kind of like if you ever see two individual soap bubbles, all right, floating through the air, and they bump into each other, and then they kind of merge and become one. Similar type of appearance, all right? And then the other type of secretory vesicle, all right, will do the same thing. It'll merge with the plasma membrane, but in that case, its contents go outside the cell. So let me show you on this awesome slide here. All right, so here's an electron micrograph of the Golgi apparatus. Here's the cis face here, that's closest to the endoplasmic reticulum. Here's the trans face. You can see a couple secretory vesicles being formed here. All right, there's one, okay? So, our picture here, here's our endoplasmic reticulum. It's working hard, making proteins, all right? It packages up some of its proteins that it's very proud of, and it says, all right, it's time to send it off to the Golgi apparatus. So it packages up its proteins, sends it to the Golgi apparatus. We got our cis face here, which is closest to the endoplasmic reticulum. So it merges with the cis face. The proteins enter in, they get packaged up and modified and all sorts of stuff, and it gets passed from sac to sac to sac till we get to the trans face. And then the trans face will package up those proteins that it's modified and whatnot. And by then it knows where it's going. It's either going to go here, all right, to the plasma membrane, and then the contents are going to merge into the plasma membrane. Say it's some phospholipids. We got some phospholipids for you. All right, we want to add those in, okay, or whatever. Or we have specific proteins. We're going to learn about integral proteins and peripheral proteins, which for gates and channels and all that. Or now, hey, we got some stuff that we got to send out and get into the blood. All right, it's some wonderful, delicious insulin. So it packages it up, sends it into the secretory vesicle, has the secretory vesicle merge with, all right, it merges with the plasma membrane. And that process is called exocytosis. We'll talk more about that in detail. Don't worry about it now. And then it spits out and ejects its contents into the extracellular fluid or space, or we also call the interstitial fluid. Okay? Just a brief kind of explanation there as to what the Golgi apparatus does. Kind of cool. All right, another membrane-bound organelle is the lysosomes. Now, the lysosomes have a pretty cool job. All right, they're involved in a couple different things. Okay, but what they'll do is they'll help to kind of, well, and if there's junk floating around on the inside of the cell, they help to clean that up. Okay, because these structures here have these digestive enzymes on the inside of them. So say there was some sort of synthesis reaction occurring in the body, in the cell, and it just didn't get finished for whatever reason. So you have these partially made molecules, no problem. The lysosome goes around and just gobbles them up, breaks it down, all right? And then you can recycle whatever wasn't used, right? But what will happen too, right? If the cell, depending on its function, okay, takes something from outside the cell into the cell, that's called endocytosis, okay? And when that happens, all right, so essentially the cell itself, if anyone is familiar with an amoeba, the cell can shoot out these pseudopods, and it'll surround something that's outside that it wants, and it'll eventually surround whatever it wants, and it pulls it in. So the lysosome will then combine with it and digest whatever was inside that pseudopod, that vesicle there, okay? So, for example, thyroid cells, all right? They make their cuboidal, simple cuboidal cells, excuse me, and they're going to make thyroxine, your thyroid hormone, right? But in order to activate thyroxine, it needs to combine with the lysosome. So thyroxine enters into the cell, okay? And then what the lysosome will do is that it'll merge with that little vesicle that it made with the inactive form of thyroxine, and it'll undergo some chemical processes that will activate the thyroxine. And then what that cuboidal cell will do is it will eject it back out into the bloodstream or wherever it needs to go. Right? So lysosomes will help in digesting unneeded substances that were just kind of lying around, but at the same time, they'll help with digesting the contents of something that the cell itself brought in, okay? For example, the thyroxine. And then it digests part of the molecule, not the whole thing, because then it would be pointless. So it'll digest part of the molecule, and that process there activates the thyroxine, and then that cell will then expel the thyroxine out to where it needs to go. All right, peroxisomes. I'm going to let you write a few things down here while I take a quick sip. All right. We talked about the rough endoplasmic reticulum, because the rough endoplasmic reticulum makes these structures here. Okay, they get pinched off from the rough endoplasmic reticulum, but inside they have, and you want to make sure that you know, whenever you see oxidative enzymes, just think these are really powerful enzymes. Okay, very, very powerful enzymes, and you can see some of the things that they do. All right, these peroxisomes. So these peroxisomes are these sacs that have these powerful enzymes on the inside. Part of that will be, again, when we talk about chemical digestion, all right, they'll run around and break down certain molecules, fatty acids, for example, amino acids, all right, they'll start to break those down, all right. For example, too, all right, you look here, detoxification, this molecule right here, that's hydrogen peroxide. Well, when you're not using hydrogen peroxide to dye your eyebrows or your hair, it is great when you don't have bleach available because hydrogen peroxide can kill bacteria and certain viruses pretty well. So these peroxisomes make this hydrogen peroxide, right? So they can go ahead and kill certain things, right? When they're breaking down, right, certain fatty acids, that process is called beta oxidation. Because remember what I told you? You saw that fatty acid chain, right? It had all these molecules strung together. OxyClean, yeah, kind of like OxyClean. Right? You have all these fatty acid molecules stuck together, and through that beta oxidation, we just start cleaving those off, and we start to break down, right, those individual molecules to get energy from. And that's one of the things that the peroxisome does, right? But at the same time, all right, they'll also help to make certain types of lipids. You heard me talk about the bile salts there that are needed for digestion, where your liver is going to have these peroxisomes in there, and they'll help to create these bile salts, which help with the actual fat breakdown of anything that you eat in your diet. All right, that's it for the, no, we're not going to worry apart. Bam. Let's talk about the mitochondria, one of my favorites. We're going to be talking about this more later on. All right, but one of the things you want to understand is this is a membrane-bound organelle, but its membrane has a double membrane. All right, and you won't talk about the significance of that in this course. You'll talk about it in 211. Okay, but it has to do with energy production. Okay, and the specific type of energy production, all right, or metabolic energy production is called aerobic cellular respiration. Star that. That's very important. Aerobic means with oxygen. If you don't have oxygen, your mitochondria are useless, okay? Because the only way to get the mitochondria to make energy is with oxygen. If you don't have it, the mitochondria will not be doing anything, okay? So they make ATP, all right? They're not the only thing that makes it, but that's their main job, make ATP. So let me put it into perspective for you. You get one glucose molecule. Okay, one glucose molecule, when you break it down and there's oxygen present and you involve the mitochondria, you'll generally yield about 32 ATP molecules from one molecule of glucose. That's pretty good. Without oxygen and without the help of mitochondria, you'll generate about 2 ATP from one glucose molecule. So you don't have to be a math genius to figure out the advantage of having mitochondria, okay, involved in energy metabolism, okay? So their nickname, the powerhouse of the cell. That's their nickname. That's their job. Make energy. All right. So what organelle functions in detoxification and beta oxidation? All right, the peroxisome, we just talked about that. And then energy production, the powerhouse of the cell, that is the mitochondria. Okay, peroxisome, mitochondria. All right, I'm going to try to finish up all the organelles here. All right, the non-membrane bound organelles. All right, there's not as many, so not to worry, guys. Okay, so ribosomes are going to contain both protein and then ribonucleic acid. What is that? That's RNA. Okay. RNA. And it's the RNA that's going to help us to make proteins. Okay. Don't worry about the large and small subunit and the APE sites. All right. Don't worry about all that. But what you do want to know is that these guys are made inside the nucleolus, not the nucleus. Remember, the nucleus is the big ball, and there's a smaller ball inside of it. The nucleolus is the smaller ball. All right. So what happens is these ribosomes get made. All right. These large and small subunits get made in the nucleolus, and then they get sent out to the cytoplasm, and then they get put together. Okay. So there's two types. All right, bound and free. The bound, those are the ones that I pointed out to you earlier that sit on the surface of the endoplasmic reticulum. Okay? So it's these guys here that are going to make the proteins that are eventually going to be sent out of the cell, or they'll become part of the plasma membrane. All right, and I didn't mention this earlier, but they'll also be utilized, all right, to make enzymes for the lysosomes. All right, and it's the free ribosomes that make all the other types of proteins, which means they're just floating around in the cytosol, and they'll make proteins, all right, that are going to stay within the cell, because maybe the cell has some business that it needs to take care of. It needs to make stuff, synthesize stuff, okay? So you got the two types of ribosomes, the bound ribosomes and the free ribosomes. If you want more detail on a lot of this stuff about how DNA and RNA makes proteins, read chapter 3. But I would recommend that you don't read chapter 3 until after the course is over with. It's a great chapter, but it's just too dense for the purposes of this course. Right? Is that why you can't use your fingers if you have frostbite, because no oxygen is getting through? No. What is going on there is, if you're getting frostbite, depending on how bad it is, but usually, one, your nerves are starting to malfunction because you're messing up the nerve signal transmission. And if the nerves can't function, then the tissues are having an issue, like your muscles, your skeletal muscles can't function because the nerves aren't telling them what to do. And at the same time, you're starting to get what we call tissue necrosis, when the actual cells and the tissues will start to literally die. It does sound horrible. It's awful. And I'll talk about that when we get to the skin chapter, though. All right, so this is a picture of RNA. And the only thing I want you to really see is, not RNA, excuse me, the ribosomes. Here's the endoplasmic reticulum. You can see the bound ribosomes here, and then you can see clusters of just some free-floating ribosomes. So these are the bound, and these are the free ribosomes just floating around. All right, the centrosome. To be honest with you, the only thing that you really need to know about the centrosome is, one, it usually stays close to the nucleus, and the only time it's of any real consideration is when the cell's dividing. Because what will happen is, you have your cell here, one centrosome will be over here, another centrosome will go to the other side, and they create what's called the mitotic spindle, right? Which are these, I believe they're microtubules that they send out, and they go to the center of the cell. And what they'll literally do is they'll start to split the cell. They rip the cell in half. Yeah, it's crazy. And then you'll have two cells. I'll show you a picture of what the centrosomes look like. All right. But that's what they do. Their job is to organize those microtubules within the cytoskeleton, but specifically for the mitotic spindle. This is what they look like. So when nothing's going on, they sit on top of one another, but perpendicular to one another. And the only time they're ever separated from one another is when the cell is going to divide. Okay? For a long time, a lot wasn't known about them. All right, one of my favorite non-membrane bound organelles, the proteasome. Literally, important, note this, it's the quality control for exported proteins. So this organelle essentially digests proteins. Okay? So what happens? Because not every protein that your cells make are perfect. Nah, they're not perfect. Okay. So in a case where a cell makes an imperfect or malfunctioning protein, well, it decides, you know what? Let's get rid of this thing. Okay. So what it does is the cell marks the protein with this ubiquitin, and it tags it. So there's a little tag that goes on, and it says we need to get rid of this thing. So we have this organelle. I'll come back to this slide here. You've got this organelle. Here's the bad protein, bad protein, or it's an old protein. It just needs to go. So the cell tags it with ubiquitin, and these proteosomes float around. They're like, oh, all right, we see that this protein has the ubiquitin tag on it. So it just funnels it down through the center, and it just snaps off. All these little balls, those are the amino acids, and it just snaps them off. So then your cell can either reuse these amino acids, all right, for another protein, or it just destroys them. Okay? Pretty cool. Pretty cool. All right. So again, we don't go into enough detail about what happens when proteins are made, but there's different configurations for proteins. Some are a long strand. Some of those strands will get folded into what they call a beta sheet, a flat, like a piece of paper. And then if you crumple up that piece of paper, that's a tertiary configuration. All right. Well, if improper folding occurs, well, then we've just ruined that protein, so we tag it with ubiquitin. So these proteosomes just kind of march around and they check that stuff out. All right. So we have them both in the cytosol and inside the nucleus. Okay, because you have proteins in there too, and they're just constantly patrolling. All right, which non-membrane bound organelle functions primarily in digestion within the cell? Proteosomes. All right, that's what they do. They march around. They're the quality control expert on proteins. Okay. All right, we're going to stop here. Woo! That's a lot to take in, you guys. I still got many more slides to go, but we're not bad. We're doing good. We're doing good. All right. So listen, I'm going to stop recording here. I know mine hurts.